cells communicate with each other, using the action of the hormone epinephrine to frame the discussion. Recall that epinephrine, produced by the adrenal glands, is responsible for the so-called fight or flight response, a coordinated set of physiological responses brought on by being startled or scared or otherwise stressed in a short-term fashion. The specific aspect of this response we were interested in was the effect of epinephrine on cells in the liver, because epinephrine causes those cells to convert glycogen into glucose, which is then released into the bloodstream. In this context, then, epinephrine is a kind of cellular signal sent by adrenal cells to liver cells, telling the liver cells to produce more glucose. Now, we saw last time how epinephrine, like most external signals affecting a cell's behavior, can only exert its influence on the cell indirectly by interacting with a receptor protein that spans the cell membrane. We also saw how the interaction between epinephrine and a membrane receptor leads to changes on the inside of the cell, thus getting the message from the outside to the inside. In today's lecture, we'll follow this signal further into the cell to try to understand how a signal ultimately changes the behavior of the cell that receives it. Actually, when boiled down to its essence, this process is really relatively simple. As we saw last time when we looked at how G-protein linked receptors work, there's simply a sequence of interactions between a series of molecules, with a change in the activity of one molecule causing a corresponding change in the activity of the next molecule down the line, until at last the signal reaches whatever molecule it is, usually a kind of protein, that causes the desired change in the cell's function. So this is again reminiscent of the nursery rhyme, The House That Jack Built, or perhaps you might think of it as a line of falling dominoes. We call this sequence of events a signal transduction pathway, or alternatively, a signal transduction cascade, because the signal originally received by the cell is transduced from one molecular form to another as it progresses through the cell to reach its final destination. After we look at how signal transduction cascades work in general, we're going to conclude the lecture by looking again at the final stage of cell signaling, which is the functional response of the cell, by considering ways in which the cell can possibly change what it's doing in response to whatever signals it receives, both in the short run and in the long run. Before we pick up on the signals trail, I want to give you just a little more background on how liver cells respond to epinephrine. The idea here is to help us keep in mind where we want to get to in the end by going back and looking again at the example that we're using to frame the overall discussion. Now, I told you last time that glycogen is a storage form of sugars in cells. It's essentially a long polymer of glucose molecules all bonded together. The primary response of liver cells when they receive epinephrine is to break down glycogen molecules into individual molecules of glucose. The glucose produced this way is in a useful form that can be released into the bloodstream, providing the rest of the body with a quick source of energy. Now, based on everything we've learned so far in this course, you might guess that the molecule that actually does the job of breaking down glycogen is, you guessed it, some sort of protein, an enzyme. Let me name that enzyme. The enzyme that catalyzes the breakdown of glycogen into glucose is called glycogen phosphorylase. If epinephrine is responsible for liver cells releasing more glucose, then the molecular effect of epinephrine must eventually have something to do with changing the activity of that enzyme. So how can the activity of an enzyme in a cell be regulated? This is really turning a little bit to this question of response for a moment. Now, one way might spring up into mind immediately based on our discussion of gene regulation several lectures ago. It seems likely that one way that epinephrine can change the activity of glycogen phosphorylase is by regulating the expression of the gene that codes for that enzyme. If more of the enzyme is synthesized, and therefore more of the enzyme is available in the cell, then of course the activity of the enzyme in the cell will increase overall. We'll see this does indeed happen. There's another possibility, however, that we haven't discussed in any detail yet. This possibility involves turning on copies of the enzyme that are already present in the cell, but are in an inactive state. In fact, this occurs as well, as we'll see shortly. Now, let's take a look at how we can turn on and off the activity of an enzyme. Well, before we look at that, we might ask, why should we do that at all? I mean, after all, when we were talking about the control of gene expression a few lectures ago, I made the point that it's expensive to build proteins if you don't need them. So ordinarily, I argued, a cell should only produce a protein when it has to have that protein for its function. So why should we worry about having proteins, enzymes that are holding in an off state and being able to turn them on? Why not just always produce more enzyme when we need it? The reason is that although it's expensive to build a protein, it also takes time to build a protein. For example, when E. coli bacteria are exposed to lactose, it takes at least a couple of minutes before the lactose metabolizing enzymes we discussed appear in any concentration in the cell. Now, a couple of minutes may seem like quick work, and it's certainly fast enough to take advantage of the lactose that an E. coli might want to use as a food resource. But in other cases, a couple of minutes is far too long for a useful response to some signal. This is certainly true for the fight or flight response, where the response to an immediate challenge must be measured in seconds or less, not minutes, if it's going to be adaptive. Gene expression simply takes too long for many of the activities that must be turned on and off in cells, and in these cases, other mechanisms have evolved to turn proteins that exist on or off. Now, one particularly important and actually quite ubiquitous way in which the activity of proteins are switched on and off involves the addition of what's called a phosphate group to the amino acids to one of the amino acids in the protein through a process called, not surprisingly, phosphorylation. So a phosphate group is simply a chemical group that includes a phosphorus atom at the center bonded to 4 oxygen atoms. We've seen this chemical structure before, actually, when we looked at the structure of nucleic acids. Remember that the chemical backbone of a single strand of DNA is an alternating series of deoxyribose sugars, a sugar molecule, with phosphate groups, this phosphorus atom bonded to oxygen. Phosphate groups are highly negatively charged. They carry with them a very strong negative charge. So when they're added to a protein, that charge has a particularly profound effect on the protein's shape, which causes, of course, a major change in the protein's ability to function. Many types of proteins will assume 2 different shapes, 2 different conformations, depending on whether or not a phosphate group is attached. The protein will typically be in an inactive form when there's no phosphate attached, and then the attachment of the phosphate changes the protein to an active state. In this way, the addition of the phosphate serves as a kind of chemical switch, turning on and off the protein's activity. Indeed, many kinds of enzymes have as their function the addition of phosphate groups to other proteins. That is, the reaction that these enzymes catalyze is the reaction that adds a phosphate group to yet another enzyme or some other type of protein. The general name of an enzyme that phosphorylates other proteins is a protein kinase. The importance of protein kinases for the function of a cell in general is absolutely enormous. A single cell may have hundreds of different kinds of kinases. And in fact, one estimate suggests that about 1 out of every 100 genes in a eukaryotic organism's genome codes for some sort of protein kinase. So you can see they're very important. Now, the sequential phosphorylation of a sequence of protein kinases, with each protein phosphorylating another kinase in turn, is an especially important mechanism underlying signal transduction cascades in cell signaling. So let's take a look at how this works by getting back to the question of how epinephrine ultimately affects the activity of liver cells. We left off last time with the action of the G protein receptor system causing the production of a molecule called cyclic AMP by an enzyme called adenylyl cyclase, which occurs on the inside surface of the cell membrane. The cyclic AMP molecule, in turn, is what triggers a series of proteins to sequentially phosphorylate each other. That is, to turn each other on in a sequence, in a signal transduction cascade. For example, in the cascade that is involved with epinephrine signaling, the cyclic AMP causes an enzyme called protein kinase A. Pretty simple name, it's a protein kinase, and it's the version A, to become phosphorylated. So protein kinase A becomes phosphorylated through the action of cyclic AMP. What happens then is protein kinase A in turn causes yet another enzyme, this one happening to be called phosphorylase kinase. These kinase names do get a little confusing, but it causes this enzyme phosphorylase kinase to become phosphorylated itself. The phosphorylase kinase then in turn phosphorylates the last enzyme in the sequence, which is glycogen phosphorylase. And remember, it's glycogen phosphorylase, which is the enzyme that is actually responsible for producing glucose by breaking down a molecule of glycogen. Now, as I said earlier, if we don't worry about memorizing all the names of all the kinases involved, there's nothing really conceptually complicated about how a signal transduction cascade works. It's just a series of proteins activating each other in sequence like falling dominoes. But then you might ask, what's the point of having a cascade? I mean, after all, why doesn't cyclic AMP just go and turn on the glycogen phosphorylase itself? Why go through all of these intermediates? It would certainly seem more straightforward. Actually, as it turns out, there are several advantages to sending the signal through a series of intermediates before it reaches its final target. The first of these advantages has to do with the fact that the signal becomes extremely amplified by the cascade. Let me explain what I mean by that. Each individual enzyme molecule, each kind of enzyme in the sequence that's activated at one step in the pathway can itself activate many, many molecules that occur at the next path. That is, once one of these kinases is turned on, it can phosphorylate more than one of whatever other kinase it happens to work on before it itself gets turned off. As a result of this, the strength of the signal cascade increases dramatically with every additional step. Just a few molecules of cyclic AMP at the start of the cascade can result in the activation of a huge number of glycogen phosphorylase molecules at the end. If you include the steps that lead up from the original binding of epinephrine to the G protein receptor complex we talked about last time and add that to the pathway, which is really part of the whole cascade, then one theoretical calculation suggests that a single molecule of epinephrine binding to the cell will cause on the order of 10 to the 8th molecules of glycogen phosphorylase to be activated. That's a ratio of 1 to 100 million. So you can see the power of amplifying signals, amplifying the effects of the signal through these cascades. A second important consequence of having a signal cascade that includes several intermediate steps is that the activation of different proteins along the pathway can lead to multiple functional outcomes. Let me explain this. Let me explain this by example. For example, one of the early steps in the phosphorylation cascade that starts with cyclic AMP causes another enzyme called glycogen synthase not to be turned on, but instead to be turned off. The same step that leads to the activation of that protein kinase A at the beginning of the pathway that eventually turns on the glycogen phosphorylase will itself turn off glycogen synthase. What's the significance of that? Well, it has to do with the activity of glycogen synthase. As its name suggests, glycogen synthase is an enzyme that actually synthesizes glycogen. In other words, it takes glucose out of the cell and builds up more glycogen. So it has the opposite effect of the glycogen phosphorylase. glycogen phosphorylase releases glucose, glycogen synthase takes it up. It makes sense that if you want the cell to be producing more glucose, you should both turn on the enzyme that produces glucose and turn off the enzyme that takes glucose up. And that's only achieved through the coordinated activity of these two enzymes because they're both interacting with different parts of the signaling cascade. There's another important functional consequence of signaling cascades, and that is that they allow for crosstalk between different pathways and thus allow for more control and modulation of the cellular response. Many of the kinases or other kinds of molecules that are involved in signal transduction pathways are associated with not just one pathway, but a whole series of different pathways. That is, these molecules may be shared among many pathways, which means that few, if any, of these signaling cascades are simple linear paths. Instead, these cascades function more like networks, with multiple inputs as well as multiple outputs. The fact that signaling cascades interact in this way within the cell make it possible to integrate the effects of several external signals, as well as coordinate these effects with other ongoing cell functions. For example, epinephrine triggers the production of glucose from the breakdown of glycogen, but there may be other factors that should equally well influence how much new glucose is produced. It's possible, for example, that blood sugar levels already are very high when an individual suffers some shock leading to the release of epinephrine. It's still adaptive for the cells in the adrenal glands to release epinephrine because epinephrine has other jobs to do in the body. Other adaptive responses are modulated by this same hormone. But the cells of the liver don't need necessarily to release glucose if there's already more than enough glucose available. In fact, it may be maladaptive for them to do so. In this case, other signals impinging on these liver cells from other sources that reflect the current status of blood sugar levels can adaptively modulate the effects of epinephrine on glucose production. And this happens again because these signaling pathways are occurring in interacting cascades. Conversely, there may be other kinds of signals from other sources that also trigger the production of glucose. I mean, it's possible that not only epinephrine triggers production of glucose, but other signals from other sources adaptively should cause the glucose to be released. And in this sense, they would enhance any effects of epinephrine if it's present as well. In general, the interaction of kinases and other signaling molecules in the cell provides a mechanism for an intracellular integrated response to signals on the molecular level, an integrated response that would be more adaptive than each of the individual responses occurring on their own in isolation. Well, I've emphasized the role of enzymes and specifically protein kinases in signal transduction cascades, but there are a few other kinds of molecules that also play particularly important and general roles in signaling inside the cell as well, and it's important to point these out. These molecules are not proteins at all. In fact, they tend to be very small, hydrophilic molecules, very specialized, or in one case, even individual ions, maybe several cases. We've already encountered one of the most ubiquitous of these molecules, which was cyclic AMP. Remember, cyclic AMP is produced by the enzyme adenyl cyclase in response to the binding of a G protein. And this was the first element in the signal transduction cascade that led to the activation of the enzyme glycogen phosphorylase and thus the production of sugar. In this position, cyclic AMP represents the first intracellular signal, the intracellular signal that is most closely related to the extracellular signal epinephrine. As such, we refer to cyclic AMP as a so-called second messenger. The name second messenger is given to this kind of signaling molecule as a contrast to the external signal, which is thought of sort of as the first messenger. Second messengers play an especially important role in intracellular signaling because they often interact the most broadly with the most different kinds of signaling cascades. I've mentioned already cyclic AMP is chemically related to the nucleotide adenine, which is adenine, I should say, which is one of the nucleotides found in nucleic acids such as DNA and RNA. Two other very important second messengers found in cells are actually small molecules derived from lipids. These are called IP3 and DAG for short. I won't even bother giving you the long chemical names of these molecules. The point is this. Like cyclic AMP, these molecules are usually the first intracellular signals generated in response to activity of an extracellular signal. And, like cyclic AMP, they have widespread effects on signaling pathways. Actually, the last second messenger I'll mention is very interesting because it is a calcium ion. Calcium ions are particularly ubiquitous as second messengers, affecting many, many aspects of cell function. Now, calcium ions present us with an interesting puzzle when we think about how second messengers or cell signals can work. Calcium ions are just simply ions. And in fact, they're everywhere. Well, they're not exactly everywhere, but they're not the kind of molecule you would think would be involved in some complex signaling pathway. They're just ions. It turns out, however, that cells regulate the amount of calcium ions inside of them quite closely. There are specific mechanisms by which calcium is removed from the cell or taken up into compartments inside the cell. And these mechanisms, therefore, keep the concentration of calcium ions very low inside the cell. If the cell is going to use calcium as a signaling molecule, what it does is essentially open up channels, floodgates, that allow the calcium to rush into the cell. And because calcium is in a very low concentration in the cell and might be sequestered in very high concentrations elsewhere, it rushes in very quickly and perfuses the whole cell almost immediately. And in that sense, it can have a very broad influence over the cell in a very short period of time. In fact, the way calcium ions interact with other enzymes is by collaborating with a protein that is the protein that actually does the interaction with other enzymes or other proteins. This is a protein called calmodulin, which changes its configuration in response to the binding of calcium. So calcium acts as the second messenger, just an ion coming into the cell. It binds with calmodulin, and calmodulin will then go off and bind with some other protein. Well, let's turn back now at the end to the question of how the cell responds ultimately to the signal. As we've seen, the response of cells to signals involves either turning on or turning off the activity of proteins already present in the cell, with this mechanism of phosphorylation being a particularly important way to turn on and off proteins, or it involves turning on or off the expression of genes that will result in the synthesis of new proteins in the cell. The production of glucose that we've discussed actually represents an example where both mechanisms occur. There is a store of existing enzyme, the glycogen phosphorylase in this case, in the cell, normally in an inactive resting state. The signaling cascade that we've discussed has, as one of its final steps, the phosphorylation of this enzyme, which turns it on and therefore makes it possible for this enzyme to begin to synthesize glucose by breaking down glycogen. But there's another branch to this pathway. This same signaling pathway will phosphorylate a protein that in turn goes to the nucleus of the cell, binds with another protein in the nucleus, and when bound with that protein will go to the DNA and act as a transcription factor. This transcription factor, when it's activated, will be a transcription factor that helps turn on the gene for glycogen phosphorylase. And in this way, more glycogen phosphorylase is produced, in addition to some glycogen phosphorylase being turned on. Now, what's the difference here? Well, as I said at the beginning, the major difference really is one of time. Turning on and off existing glycogen phosphorylase can occur very quickly. But not only can it be turned on very quickly, it also essentially can be turned off very quickly. It has a short-term onset and also potentially a very short-term offset. Turning on the expression of genes that control the production of more of the enzyme takes longer, but it also has a more lasting effect. Once the protein is produced, it will stay present in the cell for some period of time. So epinephrine, in this sense, has two very different ways of changing the behavior of the cell, switching on and off existing proteins and building more proteins. Now, there's one more point I want to make about cell signaling, and it's this. A cell will respond to a particular signal, such as epinephrine, depending only on whether it has the receptor for that signal present on its membrane. In other words, liver cells respond to epinephrine because they have a G-linked protein receptor that is specific for epinephrine. Other cells in your body, for example, cells in your pancreas, may not respond to epinephrine. Well, why don't they respond? Well, it may be that that particular organ has no important role to play in this fight or flight response or anything else that epinephrine is involved in controlling. Why don't those cells respond? The epinephrine is present at those cells. When epinephrine is released into the bloodstream, it goes throughout your body and it will potentially interact with every cell. But those cells that don't respond are not responding because they don't have the receptor for epinephrine. Now, there's a flip side to this story. It makes sense that some cells should respond and some cells shouldn't, and the way that they get sorted out is whether they have the receiver or not for the signal. But two different cells that both receive the same signal could have two different responses. Remember, epinephrine not only causes liver cells to release glucose, it causes cells in your heart to cause the heart to beat faster. These are completely different functional responses. They have nothing to do with each other than the fact that they're responses to the same signal. How does this happen? Well, the way that this happens is because the receptors both respond to epinephrine, but lead to different signal cascades that eventually lead to different functional responses of those two kinds of cells. So the point here is the fact that different cells share some types of proteins in common may have something to do, or really does have something to do, with the way we can get coordinated responses that are very diverse to a common signal, coordinated responses that are adaptive, such as the coordination of responses involved in the fight or flight response. Well, over this lecture and the last, I've outlined the mechanisms through which cells talk to each other, focusing on the mechanisms cells use to receive, process, and respond to signals. These lectures have introduced a number of details, but the specific details we've covered here are not really as important as the general principles they reveal. We've seen the importance of several general mechanisms of cell function that are necessary for cell signaling to occur, and we're going to see these same mechanisms over and over again later on in the course. What are some of these general principles? One is that ligands bind to receptors, and this is the way something happening on the outside of the cell can affect anything happening on the inside of the cell. We'll see this again and again. Another thing that we've seen before and have seen now here has to do with the importance of shape changes in proteins regulating their function. What we've introduced today is the idea that phosphorylation by protein kinases is a particularly useful and general way that this shape change occurs. We've also seen that signal cascades can lead to the amplification, modulation, and integration of signals, and we will again see that again and again. We've only talked about one example of a signal, epinephrine, but of course there are many, many other signals that are involved in the way that organisms work. In fact, these principles of cell signaling that we've discussed will become very important to us as we begin in the next lecture to look at how organisms develop a bunch of different cells from just one single common cell.